biology, and it's a good aspirational guideline set for collecting them for publication processes. To be fair, I've never actually met all those requirements that are listed in this 2017 update on publication standards. It is very onerous, but at least it's an aspirational goal, so you can see the kinds of things that you would want to report in a publication. One of the couple of things that you'd want to report, or they're asking you to report, are the wavelength that you collected at, which again doesn't really matter because it's intensity versus Q, and Q takes care of the wavelength. But you'll put it in your manuscript. You collected it at 11 kV, which is the equivalent of 1.127 angstroms. We collected for a full 10 seconds on your samples, but they were framed at 0.3 seconds. So that's how you ended up with 33 plus frames for every sample. So basically the shutter's open, exposing your sample for 10 seconds fully, but the camera's taking snapshots at a 0.3 seconds, and then going to the next frame, and so on. And so you guys are probably wondering how to deal with that data set, and I'm going to show you how that's done. I'll go through a data set. But moving on quickly, we've made a lot of progress on our end in terms of data quality and success of data collection. So what we've done is we've introduced a new system for collecting, where we've made the needles on a robot X-ray transparent. So we're pulling up your samples into a liquid handling robot needle. The needles go in front of the X-ray beam. It gets exposed, sample gets dumped back into your sample cell, and then we can wash with system fluid. So we pour 1.5 mL through that needle to wash out what's there, and then we literally go to a hair dryer and dry out the samples. So that is, when we get it fully implemented, going to allow us to do something like 100 samples in 15 to 30 minutes. So right now we're sort of at we're just using a single needle. We're perfecting our single needle so that we can go to ×4 eventually. So right now we're about 2.5 hours per plate. So we're really aiming for high throughput. As you know, we also required 30 microliters. We're hoping to eventually go down to 10, but we're not there yet. So those are on the horizon, and so get yourselves thinking about what you could do in high throughput, like constructs, drug screening, things like that. But this development hasn't come without its problems, and we've encountered 3 major problems, and I'm going to go through them, and I'm going to ask you guys to look for them. The first is a high Q problem. So what I'm showing here is the scattering from one sample, where the buffer before the sample was subtracted from the sample, and the buffer after the sample was subtracted from the sample. And those result in 2 independent curves, a black and a red. And so you can see that there's some deviation there. That turned out to be temperature cycling inside the hutch. So just be aware, take a look at that. Make sure that's not happening. If it is happening, I mean, the most obvious thing to do is to average the 2. However, sometimes it's possible to tell which one's correct just by looking at the decay. In this case, the black one is correct. We also had and have had bubble problems, and I think Svetlana samples had some issues here, which we fixed by washing at every sample. But we supply a image of every sample inside the needle. Full should look like the top one, empty should look like the bottom one, and you will be able to scan through them, and I'll show you an example in a moment. But this is a picture of the needle, and we just have bubbles sometimes creeping in, or it's not picking up properly. The final issue is a small Q problem. In the very small angles, small Q problem, you might have some deviations. Again, the last couple data sets, pretty much everything's been perfect, pretty much, or perfect relative to what we had before. But look at your small q problems before, differences between buffer 1 and buffer 2. Make sure they're equivalent. In this case, there's a disagreement. The most obvious thing to do is to clip the data back. Next thing to do would be to average the two. And then the other possibility is to decide which one's correct. And in this case, it's obvious which one's better. It's the black one. It smoothly goes up to small q, and there's no discontinuity. So you have to make some decisions in that case. And these are just the needles, buffer 1, sample, and buffer 2. And you know, we're still working on why the small q would be happening. It's like maybe some liquid on the surface of the needle or something. So we're working through that. So now I'm going to go through a data set. All right. Are there any questions about that collection process?
No.
All right. Cool. Let me move over here to I've collected a sample for UCSD. Sample pics look like this. I guess I can let's see, view, and then you can basically even you know, use whatever viewer you can to just screen through the images, or you can click them one by one, and just to make sure that there's no bubbles in any of the samples or buffers, so you can look through that. Back. So your data sets, when you look in your directory, you have your results in the subtracted folder. There is also possibility of getting the unsubtracted data as well. And that means buffer 1 or buffer 2 have not been subtracted from your data, so you'll get everything, including buffers and samples. So that allows you to subtract on your own. We've done this for you, and we've done it. We've scaled the buffer and the sample according to our diodes of intensity for the X-rays. So we think that does a pretty good job. But in each directory, you'll also have your buffer 1 subtraction, your buffer 2 subtraction, and your average. Within your buffer 1 subtraction, you'll have your 33 frames that were collected. So let's open up a tool here to start processing that data. Let's see, what do I want to pull up here? Let's go here. All right. So I'm showing the news, but this is really what I want to show you. There's a web tool called at SIBYLS.als.lbl.gov/ran. So that's on the web. You don't have to download anything. It's totally available to use, and we recommend you using it. There are other tools available that we haven't provided, so feel free to try those other things, but here's one way of doing this. Another little warning is that Catherine goes through your samples already once, and when she does that, sometimes she'll create a, I guess for biologists can understand, a scar of her going through it, which is that there might be an AV data set within your structure, which you probably want to get rid of because you don't want to average her already average sample in there. The way I do that is to do like a search for 00 AV. That would be kind of, in this case, there isn't any, but just make sure there aren't any of those in your structure, in your directory structure. So I'm going to go to this first one. I'm going to pull buffer 1 down, and you just drag the whole folder over. And again, you guys are probably mostly Mac. How you drag and drop is a little different, but you can drag the whole folder in. And so this is a scattering curve. It's the first low concentration of the sample. And it's separated into 3 different regions: a Guinier region, a Porod region, and a wide Q region. The tool allows you to zoom in on the different regions. And what we're looking at right now, what is appearing should be a dark-ish black curve, which is only the very, very first exposure, only the very first 0.3 second exposure. The orange one in the background, that's using all data sets, all averaged together. The extent to which they disagree with one another is indicative of how much radiation damage it's received over that exposure. So to the extent that the orange line doesn't fit within the noise of that first exposure, that's radiation damage, and that's what we're going to correct for right now. So the way I do this is I'll hit the Guinier button first. That'll zoom into this green region. So I hit Guinier here, and so that's zooming into this small Q region. And and you can see the deviation between the dark teal and yellow and orangey curves. So I'm going to start adding frames on, so leading up to that 33, ultimately 33. But as I add frames, let's say I add 13 frames, I'm going to ask myself, am I within the noise of the first exposure? And the first exposure here is the light blue curve in between. So it looks like the teal curve is beyond the noise within the data. It looks like out in the sort of wider part of this Guinier region. It's no longer within the noise. It's also deviating in the small Q region. So I'm going to backtrack that, and I'm going to go down to 4. Even there already, it's still not quite within, so I'm going to backtrack a little bit more. 3, maybe 3 frames already. Looks better. Maybe 2 to be safe. Okay, so this sample is very radiation sensitive. So I'm going to have to be extremely careful when I process this so I don't lose information due to exposure. All right, so then the next region is the Porod region. And there's this little scale bar here at the bottom, which allows me to drag a little bit over. And so this delineation between green and yellow is where the Guinier region ends and the Porod region begins. And let's say I add all 26 frames here. You can see that a discontinuity develops between the Guinier region and the Porod region. There's just this drop here between the two. And also the Porod region does not fit within the noise of the data. So I'm going to back off, obviously. 18, still no good. 13, still no good. 9, still no good, but better. 8, still not quite right. 4, it's getting closer, but it's really close, right? So this means that the number of points I can take in the Guinier region and the Porod region are quite similar. So that means that I can use this just 2 frames from the from the Guinier region, and I can extend the Guinier region further out. So let's say I go out to 170 points. Okay. So now I'm just using 170. So I've extended the Guinier region, and I'm just taking 2 curves within that region. So now I'm going to look a little bit further here, and I'm going to add frames, and now I'm going to zoom in from the other side here. And now I can use 6 frames sort of in this area without having radiation damage. So there's basically 6 parameters that you can play with in this GUI. There's a number of frames that you have in the Guinier region, number of frames that you put in the Porod region, the number of frames you put in the wide Q region, and how big those regions are. So let's say I'm happy with 6 in this area. I'm now going to look at the wide Q region, so I can actually just click wide and crank that out. And it looks like I can use quite a bit of the data in the wide Q region. I can use like 27 frames in the wide Q region here. That's good. Let's extend this a little bit here. And then you can see as the porod region and the there may be may be able to use more data in parts of this yellow. So I'm going to just purge this over a little bit. Again, I'm looking to be within the noise of the light blue, and I'm just looking for when I'm starting to go out of it. So it might be quite narrow somewhere, something like that. Okay, so then I have my full set. I got my full scattering curve. I've done the best I could with the different regions. I'm using first 2 frames in the Gunye, 6 frames in the porod, 27 in the wide Q. I can download this result by hitting download. All right. There is, in the package we sent you, some guidance on at least parts of this process. So, yeah. There's an automated script we created. And it's this stats RG B1 versus B2.html. So I'm going to click on that and open that up. And again, this is a totally automated script. I'm amazed that it's helpful. It's something I wrote. So again, take it all with a grain of salt. You're going to do this on your own, but it gives you some guidance. So for this B2 result that we were just analyzing, the number of files it's recommending for the Gunye region is about 2 for this first buffer 1. So it's about what we picked. It gives you a radius gyration roughly. Because the data is noisy, it really doesn't know whether it's an aggregate or not. It's a little less sensitive. It thinks that the number of points that you should use in the RG is about 66, the first 66 points. It looks at the agreement between buffer 1 and buffer 2 and decides whether there's agreement or not. So in this case, it says yes. And so anyway, there's some guidance here on these steps. So closing that further. So that's how you would create your primary scattering curves to work with. And that would be buffer 1. And then I'm going to pull up scatter here. Desktop. And I've done it sort of Martha Stewart style, where I've gone through this sample and done this for the whole data set. So if you don't have scatter, it's freely available. I have to make sure I open it with Java, and it wants the latest Java. So if you have trouble opening it, it's probably because your Java is out of date. So I've done this for the low concentration, the medium concentration, and the high concentration buffer 1, buffer 2 for this whole sample set. You can drag and drop this whole thing in there and look at your essentially 6 scattering curves for the for that process. And again, you're looking for agreements in buffer 1 and buffer 2. So just making sure things worked out all right. So let's look at the most sensitive sample, which is low. So here's buffer 1, buffer 2, and there's some disagreement here. But again, it's sort of the noise area. Looking at the high concentration, which is going to be the least sensitive to this kind of problem, here's the level of agreement. What popped out when I was doing the high concentration is this bump. And this bump is radiation sensitive. And what you want to do is you want to preserve all the bumps that you can. Those are where your structure is. And those are the things that are most likely to disappear when radiation damage occurs. So really pay attention to whether features start to blur away as you start adding frames. and back off on them. So in the low concentration, you really can't see that bump very well. It's just in the noise somewhere, but it comes out nicely in the higher concentrations. So that's a nice place to pause and ask for questions. I'm going to push further in the analysis from here, but this gives you, up till now, how to get your primary curves. Any questions?
I had one.
Mm-hmm.
When I was using frame slice, I was a little hesitant in including like all of the frames in the wide region and only a few in the Grenier. I was worried that like radiation damage might be influencing more in the wide region. That it's because the noise is so big on the first frame that it's hard to tell if you're influencing the data by adding more in. I wondered if you had encountered that at all.
Yeah, so radiation damage in SAXS is an aggregation phenomenon. So basically what happens is the charge on the surface of the protein, something gets charged there, and then that starts to aggregate the protein a little bit and making sort of bigger things. But the internal structure generally stays the same. The small Q region is sensitive to sort of this aggregation phenomenon. Whereas the wide Q region is looking more on the internal folds. So often what happens is the internal fold stays the same, the wide Q stays the same, is more radiation hard, whereas the low Q, you're starting to get these aggregates and multimers that are affecting that. So it's totally okay to do what you're doing. There are a few exceptions. If you have like an iron sulfur cluster, some kind of metallocenter of some kind, those, if they get charged up, can like blow apart and you lose some structure there. But generally, it's that's how that goes. Does that answer your question?
Yeah, that was definitely helpful. My other question was like, when I was putting the error bars back on, it sometimes looks a little strange because your error will be like large right on the edge of where your like parode region switched to the wide angle because you average so many more in the wide Q region. I just wondered if that was something that would like raise eyebrows or something if you were going to publish.
So the whole error bar thing does affect you in different ways. When you go for chi-square, there's an error bar term in there. And it's all pretty funny business, the whole error bars in SAXS. I mean, people say they've got it, but it's really hard to do that. I use chi-square as something to minimize on. So sometimes I'll have a really nice curve with very small error bars, and the chi-square will be like 13 or 14, and it's just because the error bars are so small that number is so big. Whereas if I have a really noisy curve and the error bars are huge, I'm getting chi-squares of 1 or better. And that's just because the data quality is so poor, it can't give you that sense. There also is a difference in the way error is calculated for different regions. So when you're only averaging 2 curves, it doesn't make sense to calculate a standard deviation based on the intensities in those 2 curves. It's just two points, basically, that you're averaging together. So you can't come up with an error bar based on the averaging of frames together. And what happens there is the error bar comes from sort of shot noise on the detector. But once you have 5 or more frames, you can start relying on the standard deviation between the curves. And that error bar comes from the deviation from in between the curves.
And that's all automated in frame
That's all automated, or is supposed to be. Again, there are bugs, and people have complained about exactly what you're saying before, and I probably need to, go through that error bar process again. But if I were to give you some advice, I'd probably just assume, if I were having trouble with error bars, I'd just assume like a 3% error bar across the whole thing.
Yeah.
and just apply that. And for most of what you're doing, that will work.
Mm-hmm.
And you won't have to worry about these discontinuities or whatnot. You know, and really the error bar is the deviation between the points, right? So if I zoom into, you know, this sort of noise that you're seeing on the screen, that's really the error bar. So when you plot your data with error bars, it should be on that scale.
Yeah.
Any other questions?
So I have a lot of deviation in the Gunye region for my average data and like first couple of frames, like really a lot. So because my sample is so sensitive.
Right. Yes.
So I think I was like not doing the right thing. So I was taking like first 2 frames for the Gunye region and then like all the frames for the other regions, and there was like discontinuity. But I guess it's important to keep like this like Gunye continuity, right?
Yeah. So you
know.
Yeah, you're going to Fourier transform the data to create a P-var function, and any discontinuity will create a huge amount of noise in your Fourier transform. So you really want to work on having really smooth transitions between the regions. You got to make sure that that happens.
Okay.
And remembering in your sample, because I remember looking through it with Catherine, yeah, you have your sample is quite sensitive, and that's fine. You know, and what I saw mostly was that you could take the first 2 frames most of the time, and it would still be fine. You can also sort of see how quickly the radiation damage occurs, so you can see how much. So let's say you have your frame 1 and frame 2 are slightly different. Well, then you can extrapolate from that what frame 0 would have been like, you know, the effective sort of change because of radiation damage. I would pretty much, from going through your samples, it doesn't look like it's a gigantic step between 1 and 2. So
yeah,
I think you're pretty good.
Pretty similar.
Yeah. So I think you're probably good. Next time we collect your sample, if you could just tell us, you know, I got radiation sensitive samples. Instead of framing at 0.3 seconds, we'll frame at 0.1, and you'll have like 50 frames or something like that instead of 33 frames. And that way you'll be more confident that radiation isn't as damaging. Yeah, anyway, just let us know, and we'll do it better on our end as well. Any other questions?
So I'm using a different software, not I'm using a SAXS package, like the Windows app, like Primus for the analysis. And is there any advantage of using Scatter?
I use both, and I'll show you what I mean by using both. There are a couple things in Scatter that are really nice, and there's a couple things in SAXS that are really nice. So, yeah, I think you'll. I recommend using both, or even and trying new software all the time, because this whole thing is changing all the time. So the nice thing about Scatter is that it looks the same on PC, Mac, whatever. I mean, just for teaching purposes. So the way it looks for you on this, for me on the screen, looks the same for you, regardless of the platform. Whereas with SAXS, it's written based on Fortran and has different Windows systems, so it looks different. But I'll be working in Windows in a second, and I'll show you some of the things you can do with Scatter. That you might not, why I use Scatter instead of just SAXS altogether. So that might answer that question, what you can do. Any other questions? All right, moving on.
Can I have one question?
Sure.
So in that preliminary assessment that you sent with radius of gyration already estimated, like when I go and try to estimate it with like HT-SAXS, by default, it takes less points than suggested in your app in the Gunia region, so it's a different radius of gyration. And it's kind of hard to tell when to like stop because the residuals look pretty good still, even you go further, like if it's 57 or like 67, like I don't know, it's just like how do you make the decision where to stop?
So do you use the auto RG thing, or do you use Gunia button on Primus?
I go to analysis and radius of gyrations.
Okay, so it probably sounds like an auto thing.
Yeah, auto thing. But you can vary the range.
Okay. Yeah, so I mean, you know, you want to be, so radius gyration from SAXS should be good to within 1 angstrom. So that's one rule of thumb. I'll show you what I do here in Scatter. I'll briefly go into Primus and probably do the same. So yeah, let's do it that way. Any other questions? All right, moving on here. So I have my low, medium, and high concentrations here, and I would like to compare them and see how different they are relative to one another. And sometimes there's what's called the protein likes to hang out next to itself. That's sort of like, it's not aggregation per se, but just the charging works out that it likes to be around. So that's another kind of structuring that could occur in solution. Sometimes the opposite happens, where the protein does not like itself and it's repulsive, and that sets up also a structure within the solution. And you can imagine that that kind of structuring is concentration dependent. The lower the concentration, the less each independent protein feels of itself in solution. So that's what we're doing here when we're comparing the concentrations against one another. This concentration dependence appears in the low Q region. And so the way that I deal with this is, let's just look at the first buffer for all these 3 samples here. And what I want to do is find a region where the structure, this extra structure, it's called the extra structure factor, isn't influencing. So that would be in the small q. So you can hit in Scatter where you want the data to start. And it starts at 0.001, but I'm just going to clip it back to 0.1. Okay, here. And I'm going to end the data at 0.2. Okay. And so I just have a very thin sliver of the curve. And then I'm going to hit scale. So in an area where, you know, the structuring isn't so big, and it looks, you know, again, the low concentration noise is pretty high here. But you want to superimpose them, and you can see they should scale according to how you diluted them. So assuming that the high concentration is 1 mg per mil, I have to scale the medium concentration by a factor of 2 and the low concentration by a factor of 4. So they were just 2-fold dilutions. So that should kind of go with your concentration dependence. And then I can add my points back in and see how much deviation there is in this high Q region. Also, another trick is this how the RG changes as a function of concentration. So if there's big differences here, there might be some structure factor at work if there's a trend. And you can see that there's a slight trend here that the high concentration is 24.5, the medium is 24.8, and the low concentration is 25.3. So there's a series here, and you can extrapolate the RG to what it would be at an effective zero concentration from this sort of linear scale. But anyway, you're looking for this concentration dependence, and there are ways of correcting for that. And I think if you look up on the web, I'm not going to go through that process exactly right now, but if you look up on the web, correcting for structure factor in SAXS, you'll find the process for that. So, so I've done that already, and I'm going to import my corrected form scattering curve, which is a merge of all the best qualities of every piece of data. So I'm going to drag and drop this AV here. Analysis. I'm going to unclick this. So this is my final curve with some of the best of everything. I got my little bump that I wanted to see and these things. So the first thing I'll do is I'll hit the Gunier, and I probably didn't do this quite right, but the way that you can do it here is you trim some of the data off. The rule of thumb here for a nice globular particle is that the Q times RG limit should be less than 1.3 in this last point there. So, as you increase Q, this goes to 1.6, so that's not where the Gunier approximation applies. For an elongated particle, that number is smaller. It can be all the way down to 0.08 if it's just a big thin rod. A good globular particle would be something like this. So the RG here is printed here at 24.6, roughly, for this stage here. So I'm done with that. I'm now going to estimate the volume of the protein. So in scatter, I will hit volume here. The region that I want to focus on to extract the volume of the particle is right after the bump, the first bump in the Kratky plot, sort of this slant. I want to fit that region with an exponential of some sort. Okay, so I'm going to drop the number of points here. Let's say 200. You can see part of the curve turned red. That's the last point. So I'm just going to, again, just focus in here. This bottom point here, this bottom plot here is trying to fit a exponential line through the data. And so I want to clip it at the other end and bring that up till I get something that fits a line. And you have the residuals here in this top right corner. Something like this. This P, this Poirot exponent, tells you about how folded and solid your protein is. P-value of 4 is the exponential decay of Q to the 4th. That's a globular particle. If it were Q to the 2, it would be a completely unfolded Gaussian chain. I have my volume here. It's 73,190 cubic angstroms. So that gives me now an RG and a volume, and whether it's globular or not, or flexible or not. So combining those three pieces of information, I can now calculate the mass of the particle. So I'll leave that. I'll close that out. I'll hit this volume of correlation plot, which does that calculation for me. So it takes in the RG, the exponential decay, and the volume. Does a calculation. I have to click it for it to work. And then I can go to the results tab, and the particle is about 40,000 Daltons. If it were RNA, it would be 42,000. This one's 40,000. So again, that's something you can only do in scatter, and you can't do this in Primus. So that's one nice thing. That's good. At this point, I will compare that result with the sequence and see whether this is a monomer or dimer or whatever. So in this case, I know it's 46 kD, so I'm close. I know it's a monomer, which is useful. Another thing that I will do often is look at the cross-sectional radius. So there's this RC plot. So you got radius duration, and then you got a cross-sectional radius. If it's a long tube, the radius duration will be big. The cross-sectional radius will be small. A globular particle, the radius duration will be almost equivalent to the cross-sectional radius duration. So it tells me a little bit about the protein shape. So, and what I'm doing here is just fitting these lines. So right after the Gunier region is where you measure the cross-sectional radius. I want to fit a line through this. And again, this is something you can do in Primus as well. So some kind of line through this process. Okay, and then the cross-sectional radius here is 15 angstroms. The radius of duration is 24 for this particle. So there's not quite right. So there's a very thick side, a very big side, and then there's some thinner side. So it's either a flat or a rod-like particle of some kind. Very nice, solid thing. Yes, so those are the things I would look at. I'd probably send the sequence in for disorder prediction, just to confirm, see how much disorder there is. This metapret is one way of doing that. Let's see if I got one open here. Doesn't here. There we go. So this is the protein that we were just talking about. This is the disorder prediction, and you know, the first 20 amino acids are unfolded, but maybe there's 2 domains or something. So anyway, more information. 410 amino acids, which is approximately 40 kD. So that's that. The next thing is to do a Fourier transform and pull out a P of R and a maximum dimension. To do that in scatter, you just hit this real space option here. And the default was 97, which turns out to be a very good guess for this protein. But I can clip back and go forward and backwards. And so it's suggesting that the protein's somewhere, 95 angstroms in size. You should play around with how many points you use for this process. So to make sure it's consistent, the P-var function is generally only useful when it uses up to Q of 0.35. So I'm just going to do that. I'm going to use 600 points to see how that affects this Dmax. In this case, we're pretty good. I might be able to clip back a little bit, somewhere around 88.5. And this is probably those first 20 amino acids that are unfolded and sticking out of the protein, and then you have this sort of globular region. Because there's a little tiny shoulder here, it's hard to see, but I can see that it's a little not Gaussian. This probably means it's a flattish object. You can also use this refine button, which gets rid of outliers, and so it gets rid of things that are noise for the P-var function, and sometimes changes it. So that's the P-var function. At this point, if I'm going to go for shape, this is where I go for Primus. Because in order to go for shape with Gazbohr and Damif, you need to have the output from GNOME, the HT-SAXS version of this program, to run them. So I'm going to go to Primus. I'm going to hit Tools. I'm going to find my curve. And again, for you Mac people, Primus will look a little different. Okay, so I've loaded it up. It has 1,000 points. I hit plots. It has this Gugnier button here, which does something similar, so I can back that down to 100. And get rid of those first couple points there. And the S times RG, it's up to 1.64, so I got to clip that back on the other end. Until I have 1.3. Oh, it's too far. Something like that. And the RG it's giving me here is 24.5. I got 24.6 from scatter, so they should be somewhat similar. I'm going to hit 600 here again, plot, and then I'm going to use GNOME. Be careful with how you name GNOME. It'll overwrite every time. There is an auto GNOME as well. But I noticed the particle somewhere around 98 angstroms, and I'm only going to use the 600 points. 98 here. I'm going to hit run. I get a fit to the data. You can see that the fit with 98 angstrom Dmax is poor, so it doesn't like this. And there's some messed up part here. So I want something smooth coming down. It might be here in the 80s. It might be beyond that. And when you publish your Dmax, you should publish what the error bar is. So 98 isn't quite right. Let me try something in the 80s. Still doesn't fit the data according to GNOME, and this bump is not good. So let's go to 108. Still not quite liking to fit the data. It's having a hard time. I actually haven't run GNOME on this thing, so I really want it to fit this data. So let's try 118. Yeah, GNOME's having a hard time. So it might be that I need to clip some data somewhere. Flip some of these guys back. And still not able to fit. Try the other way. All right, so at least it's smoothly feeding into the data set, into the small queue. Not quite fitting the data, but yeah, definitely examine your fits. So anyway, what I'm trying to do is I'm trying to, rather than take the whole time figuring this out right now, and what you're trying to do is have it smoothly fit into the high R for the pvar function and for it to fit the data nicely. So that's, once I have this GNOME.out, I can now go for shape restoration. There are many ways of doing the shape restoration. You can use our cluster, which has the programs available. So you can use your login for this. So if I go here to SIBYLS, Dimitri at Emble also has a server for running this, and there are also other servers around. But we've got this additional SAXS web apps here. You log in with what you have. I've got to remember my password. And then you have DAMIF and Gazbor to run. I usually run Gazbor because it's better with flexible things. You have to enter this GNOME file that we just created, a destination directory for everything to be loaded. The number of amino acids for Gazbor, not important for DAMIF, but for Gazbor you need it. That's where knowing that it's a monomer is important. The symmetry that you intend to apply. The number of residues is for the non-symmetric unit. So if you put in, you want two-fold symmetry, you put in half the total number of residues for the dimer, just the monomeric unit. You usually run 10 runs, you hit submit job, and you'll get an email with 10 runs back. So the outputs, let's see, I got an outputs. Let's see. Let's see if I can find Gazbor. All right. So you'll get something that looks like this in the end. A bunch of what's called sit files, which are shapes, and a PDB, of course. It's the average of the shapes. The important thing to look at is the average. So it will be the DAM filt. And usually the packages I end up sending or processing, this is again for a particular project where I'm highlighting how good the experiment was, what were the challenges with it, sort of the things that I mentioned already that we get, the scalars of the data, the molecular weight, the scattering profile, the P of R function, and then these shapes that you get. So this is the average of the 10 runs together, and these are the individual of the 10, how they look, how they vary from one another. You want to look at this NSD value. NSD of 0 means all the shapes are identical to one another. 1 is good, 2 is terrible. So how unique the shape is. Things I look for in quality control was whether I was able to measure multiple concentration, whether the real space RG and the reciprocal space RG agree, whether the Dmax was easy to assign, whether there was low flexibility, whether the mass was about right, and whether the Guinier region was linear. So those are things that help me decide the quality of the sample in general. So that's a very quick, rough run through, fire hose style. Any questions?
So in my case, I think I have monomer-dimer equilibrium in my samples. So do I have to go with size exclusion, SAXS, and to nail everything down?
So it depends on what you have in hand. If you have structures of your monomer already, yeah, or potential dimer.
I have traces.
Okay, that's very good. So you can go to.
I was trying to do modeling of like different fractions of monomer dimer.
Yeah.
Yeah, in SAS basically.
Yeah, so they have.
I used oligomer. So.
They have oligomer, and it's a very good program.
Yeah.
So that's one way to do it. So you kind of know. Another alternative is to do this FOX-SAXS thing, which is online. So if you do a Google search for Fox and SAXS,
okay,
you have to put the SAXS in, otherwise you end up at Fox News. You can zip up all the possible monomers and dimers and whatever trimers and whatever tetramers and things. You can put them all in one folder, zip that up, and drag and drop it onto this upload PDBs. You put in your experimental profile. You hit submit form, and it will at first come up with the best fitting structure, but then there's this multi-Fox option, which it will try to fit the data as a mixture of those things. So in the oligomer, you have to have the SAXS profiles of your models pre-computed, whereas here you can just put in a PDB and it'll do that for you. And the other thing that it will do is it'll make sure that these fitting fudge factors that Chrysol applies and Fox applies, that they're the same for all of your structures, so that there's no adjustment of the fudge factors between the different models. So it might give you a little bit more accurate percentages of the mixture.
Yeah, but
yeah, go ahead.
the problem is that probably what we see in crystal might not exactly represent what is in the solution.
Right.
So we will probably have to do some molecular dynamics maybe, and then it's probably best to have like separate data for just a monomer and separate data for dimers. So
yeah, there's two ways of doing this. You can definitely do the SEC-SAXS. That's a good way of going. I mean, that's the best way of going. You probably, in your high throughput, found the condition or construct that was most monomeric and the condition and construct that was most dimeric or larger. So those you would use to decide what condition and construct to send for SEC-SAXS. Again, for us, we're doing this high throughput thing. So that allows you to screen salts and pHs to see which one stabilizes which the best. So that's what we'd use it for. SEC-SAXS is available almost at every beamline nowadays. So we are shutting down at the end of the month, but there are other beamlines out there that you could run in that month if you can arrange it. But we'll be back in August. So.
But you also do SEC?
Absolutely.
Okay.
So the way that we run it is we are we're going to run high-throughput SAXS during the day and SEC-SAXS overnight. And so we're just opening up the application process for this, and it should be available in this August time.
Okay.
Any other questions?
What will be the application process to use the SEC-SAXS? Will it be similar to using the high throughput?
That's precisely what we're aiming for.
Okay.
It takes, so the challenge with SEC-SAXS is it takes 3 hours to equilibrate the column for a particular buffer, and then each run is at least 20 minutes. And then, of course, if you're switching columns for the thing, so it's a lot more time intensive. So you won't be able to run the variety of samples you can with the HT. But you'll be able to submit like one buffer and probably up to 5 proteins, or something like that.
My other question was using Primus versus SAXS to generate the out file. Was there a reason why you use Primus?
I haven't been able to get Scatter to give me an out file that I like. It might be that I don't have the latest version. The other reason is that it's gotten much better. Rob's made a humongous improvement in his P/R calculator. In the past, it didn't work as well, but I'm getting more and more confident with it. So have you been able to get a GNOME.out file from Scatter?
I wasn't using Scatter. I was using SAXS, so bio, raw, whatever.
Oh, right. You're using. Yeah. Okay. Yeah, you're using the Cornell thing.
Yes. Yeah, that's the one that I've used, and it seemed a little easier to modify than the Primus interface that you were showing us in terms of like messing with the Dmax and stuff. Yeah. I wasn't sure if you had experience and there was a reason why maybe Primus would be better.
Yeah, that's kind of the reason I use Scatter first for the P/R function, is because it gives me an easy way of finding that Dmax, and then I can plug that back into Primus to and get that there. Yeah, the BioXTAS thing has. I actually don't have a lot of experience. When again, that also had its issues early on, and I didn't use it. GNOME seemed to be like super solid for many years, so that's my go-to.
Yeah, I think it's a genome interface within the software.
Okay.
So it uses that in their software.
I see. All right.
Yeah. I didn't have any more questions.
Anything else?
Yeah, I'm good for now.
All right. All right. Well, thank you guys for tuning in. And hopefully, good luck with your data processing. If you guys get to the publishing stage, it's really important for us that you guys list the Beamline and our grants as where you get publishing. Otherwise, we don't get funded next year. So let us know. We're really interested in what you guys do with our data. We have a on-site workshop in October, and that's 2 days of processing data. We'll have a bunch of speakers and just work through data sets in more detail, like the shapes and modeling, and how do you process size exclusion, and how do you process 200 samples, sort of the high throughput analysis. So if you guys are interested, look at that as well. But otherwise, I'm done. Yeah. So.
Thank you.
All right. Good luck, guys.
Thank you.
All right. Take care, guys.